8. So signal transduction through cell cycle part 2. So here's
some.
And
yeah.
You guys are gonna do fine. You have practice. You have 2 exams on your belt.
You guys are all gonna be like Emma Watson, Hermione. That's your name.
And you get all of your old note sheets plus 1 new one. So remember to bring those in. Remember, they need to bring in number 2 pencil, right? For or is that a different exam?
It did. I don't know. Did Laura say if she's gonna? Did Laura say how the final exam is gonna be? Like, did she say if there's gonna be multiple choice or not? Okay, then yeah, you need a number 2 pencil.
But like especially remember to bring your old note sheets, make a new one for this one too. You are gonna be fine. And the fact that there's multiple choice, like there's gonna be some more time because I feel like there's a greater likelihood that a multiple choice question is gonna be similar or the same as something that you might have in your notes where like the open responses are a lot more open-ended. So you're gonna be fine. Like you have practice. And we're gonna prepare with I think some of the harder chapters. Like I feel like signal transduction and cell cycle people have a hard time with. So the fact that you're here, you're going to do first. We're doing some practicing.
And if anyone has any questions just throughout, just feel free to stop us.
So let's start with topic 5. The 4 pathways that we cover are RTK, RAS, MAPK, and that's a division differentiation pathway. We do survival signaling, and that's about like how the cell's default mode, I guess, is to go into program cell death. So we need our survival signaling pathway to inhibit the cell from going through apoptosis. JAK-STAT, which is involved in a whole bunch of different things depending on which JAKs and which stats you're using, but the example that we use in class is milk protein production and nutrition. And then GPCRs, and that's also, I think there are more than like 800 proteins in that family. So they also do a lot of different things, which GPCRs we have. But that's the overview of the ones that we cover. So I would kind of be familiar with like what they do, which one's differentiation, which one's, especially like survival signaling. But yeah. Okay, so RAS, RTK, MAPK, or growth factor signaling pathway. We're gonna go through the mechanism, I think, first. So just quick definitions. RTKs, they are monomer receptors.
Do you want to sit on this side?
Yeah. So our RTKs are monomer receptors. They have their extracellular binding domain, and that's where our signal binds. And then we have the intramembrane domain and the intracellular domain, which is where we have our kinase domain, our active component. And a kinase, just in case you need a reminder, is any protein that phosphorylates a substrate. Usually a protein, but not always. Sometimes the substrate might be like a lipid, which I think we've seen in one of our pathways here. And the general gist of how they work is you receive a dimerized signal. So what that means is 2 signaling molecules essentially that come together that bind to our extracellular binding domain. And that causes our 2 RTK monomers to come together and they activate each other's kinase domains. And then they autophosphorylate each other. So they're officially like activities in binding sites for the SH2 domains of their downstream binding proteins. So in this diagram, they call it the adapter protein, but we usually GRB2 the protein that binds to the phosphotyrosine on the RTK. And that binds to its SH2 domain, which is just a domain that binds to the phosphotyrosine. And the opposite side of GRB2 has an SH3 domain. You guys remember what that one binds to? It's not something that we talked about super in depth. But it'd be a cool little vocab word to throw into your open response that comes up. Go ahead. Yeah. So the protein that's going to bind to the SH3 domain is RASGF or SOS or the RAS activating protein. And it's gonna bind to, yeah, the PXXP motif is what binds to the SH3 domain. And what that is a polyproline helix. So if you guys remember from topic 1, proline is an amino acid with a cyclic 5-ring structure that connects back to its backbone. So usually it's pretty rigid and you don't see it in alpha helices. But in this case, you can have structures where prolines form a helix with themselves. And we have one of those on RASGF that binds to the SH3 domain of GRAF2. So RASGF is a GEF, and GEFs enable, right here, our RAS protein, or inactive RAS protein, to switch its GDP for GTP. It makes GDP diffuse out and allows GTP to diffuse in. And then we have active RAS. So just a reminder, RAS GEFs only work, or I guess in this case, this RAS GEF, RAS is always going to be bound to GDP or GTP. This GEF is always going to be switching GDP for GTP. That is not interchangeable with ATP. That's something that I saw in the last exam. Those are different, and it would be good maybe on your note sheet to write like which enzymes are GTPases, which ones are ATPases, just so you don't get those mixed up, because that is something that I've seen a little bit.
Well, we said about like the PXXP motif too. It's good to know because I don't know why, but it's not mentioned much like during class itself, but I've seen it come up in past exams pretty often, and it's like usually a key point that Laura looks for to like get that 1 point as part of the question. So just in case it comes up again, just to know it.
Okay, so now we have an active RAS, and RAS is going to stimulate our kinase cascade. So does anybody remember how active RAS stimulates MAPK3? Is it a kinase? Does it activate by some other mechanism? All right, so this one is kind of a key point that I think Laura wanted to drive home, especially in lecture. RAS is not a kinase. So a lot of people make the mistake that RAS phosphorylates MAPK3 and MAPK3 phosphorylates MAPK2, whatever. We don't necessarily know how RAS activates MAPK3, but it is important that you know that it does not phosphorylate it. It is not a kinase. So our first kinase in this pathway, or in this part of the pathway, is MAPK3, which then phosphorylates MAPK2, which phosphorylates MAPK, and then MAPK can act on its downstream proteins to cause some effect in the cell, whether it be division or whatever. So this pathway does seem kind of redundant. There is a vocab word for this that we talked about, I think, in topic 5. Do you guys remember what the benefit is to having these tiers of kinases? But it seems kind of just like it makes the pathway longer. So when we activate MAPK3 can go and activate 3 of MAPK2 or 5 of MAPK2 or whatever. And then all of those can go activate 5 more MAPKs. And that means that we'll have a stronger response than the signal that we started with. Do you remember the names of that? Yeah, so this provides a lot of opportunity for signal amplification, and that means that we can have a larger result or a faster result from that one signal from our one starting protein, because each layer recruits more proteins under it. All right, so unless anybody has any questions, that is the gist of RAS, RTK, and MAPK. And our next one is survival signaling. I like this one. So as I mentioned before, the cell's kind of default mode is to undergo apoptosis. We don't want that in many cases, at least. So survival signaling is just constantly activated to inhibit programmed cell death until either, the cell needs to die or it malfunctions. And when survival signaling, when the pathway isn't active or it's not transmitting a signal successfully, then the cell will die. So this is often a target for cancer biology research. Do you guys know what the connection might be between cancer and the survival signaling pathway?
If the cell can't die.
Right, yeah. So if it's overactive, when it's not supposed to be, then we will have either too much proliferation or survival of cells that are damaged and need to die. And that is kind of the hallmark of cancer, even if it's not always in the pathway. So that would be why this is a target for research. And this one, our partner to signal amplification, I guess, uses a lot of crosstalk. So that is when proteins from one pathway kind of intersect with proteins from another pathway and they work together. So we start very similarly with an RTK that receives a dimerized signal. In this case, it's going to be our survival signal. And then we have our phosphotyrosine, and we have activated PI3 kinase that becomes activated when it uses its SH2 domain to bind to that phosphotyrosine after our RTK is autophosphorylate. And PI3 kinase is kinase when it's active, it has kinase activity. So it can phosphorylate PIP2. And even though PIP2 already has 2 phosphates on it, it is still inactive. It does not function until it has this 3rd phosphate added to it by PI3 kinase. So once it is phosphorylated again in its PIP3 state, I guess, then the pathway continues downstream. And just a note about this, this is anchored in the membrane of the lipid. And yeah, I think the only one that I have on this slide. So yeah. So once we have our PIP3, that provides a binding site for AKT and protein kinase 1. So we'll have multiple of these, multiple PIP3s. We have those multiple binding sites. And protein kinase 1 and protein kinase 2 are an example of sequential phosphorylation. Do you guys remember what that is? It's kind of in the name, sequential phosphorylation. So some substrates need to be phosphorylated by one kinase before the other. And the reason for that is after they're phosphorylated by, like in this case, when AKT is phosphorylated by PK1, protein kinase 1, that causes a conformational change and it exposes the binding site for PK2. So we have phosphorylation by PK1, and that exposes the site for PK2 to also phosphorylate AKT. And then it becomes active when we are ready to downregulate this pathway. Also, the cider doesn't have it, but P10 is a phosphatase. A phosphatase is essentially the opposite of the kinase that removes phosphate groups. So P10 will turn PIP3 back to PIP2. But in our active pathway, we'll have phosphorylation by both of these in order, and then our activated AKT can dissociate into the cytosol and act on its downstream effectors. So when we have active AKT, we start off with this bad BCL2 complex. And then AKT can phosphorylate bad, and bad will then be sequestered. It can't keep BCL2 inhibited anymore, because when this complex is together, BCL2 is inactive. So that is keeping BCL2 from doing what it's supposed to do. So we need active AKT to lift bad off of BCL2. And then BCL2 can go and it binds to the mitochondria, and that is what keeps the cell from undergoing programmed cell death. So when we have active AKT to remove bad, then BCL2 is free to inhibit apoptosis. But if we don't have our active AKT, these will stay bound in that original complex, and BCL2 will be sequestered. It'll be inhibited. It can't bind to mitochondria, so we'll have cell death. And I think I pretty much covered most of this. If we have active bad, then BCL2 will be sequestered, we'll have cell death. When it's inactivated, then we will be able to inhibit apoptosis, but also like what Laura says with Goldilocks hypothesis or Goldilocks theory, we don't always want to frame the cell dying as a bad thing. So the survival signaling pathway, like what we talked about with cancer, we don't always want it to be on. It's not always a detriment to the cell when this pathway is downregulated and the cell dies. Sometimes we need that. So that's just something to keep in mind. Like we always have that happy medium. We're like downregulating things or things having poor affinity or the cell dying. Those aren't inherently bad things.
It's just,
That's just the whole pathway together. That helped me. This is how I wrote them on my note sheets instead of in pieces like the diet. Go ahead.
I'm just wondering for with AKT and PK1 and PK2. So which order, so when does AKT get activated?
So AKT is fully activated once it's phosphorylated by PK2.
And
But
PK2 has to be phosphorylated by PK1 first.
So AKT is phosphorylated by PK1. And then when this is phosphorylated by this, it opens up a binding site for PK2. So it's being
okay.
phosphorylated by both of them.
so like this part first and then this part.
So this is how it always writes in my mind sheets. I think it's more helpful to see them all together than like in the fragments that we do on the comments. And we will email these slides also after. Okay, so JAK-STAT. The example that we use is milk protein production. That's why this says prolactin receptors. But there are instances, or actually most instances of JAK-STAT, because there are so many different pathways that involve JAK-STAT, aren't going to be prolactin. That's just what we have for this example. So first, the JAKs are not RTKs. They don't have a kinase domain. They don't autophosphorylate, so they are not RTKs. That is a common misconception. That is something that I would have maybe written on your note sheet. That is something, again, like one of those little detail points that Laura likes to drive home. But the prolactin receptors. Sorry. My mouth is already in the jar like 10 minutes in. Okay. So yeah, our JAK receptor is not RTK. It is bound to our JAKs, our JAK1 and JAK2. And when our JAK1 and JAK2 interact, they will phosphorylate. And then the JAKs will phosphorylate the JAK receptors. And that is the same concept that provides a binding site for our STATs. And then again, our JAKs are phosphorylating. So our JAKs are going to phosphorylate our STATs in a different position. So they're bound to this phosphate on the prolactin receptor, and they're also phosphorylated by the JAK. And then once they have that second phosphorylation from the JAK, they will unbind from the prolactin receptor. They'll form a dimer with each other. They'll be imported into the nucleus with rich protein that we talked about in topic 4. Yeah, so we'll use important to enter the nucleus, and then they will bind to the DNA and will act as a transcription factor to regulate milk protein production. So that is JAK-STAT in a nutshell. Do you guys have any questions about the ones that we've gone over so far? What? Does the STAT like bind through? Is that an SH2 thing? I think so. Yeah. Yeah. Yeah, I think all of these. I think all the ones we talked about are SH2. That when we have the protein binding to the.
Yeah.
Yeah.
The only other one would be SH3, and then that's like with the PXXP thing.
Right. Yeah. But whenever they're binding to the phosphate.
Yeah.
Yeah. Any other questions?
Do they bind to each other through SH2, or do they bind to the kind through their receptors?
So this is an SH2 domain right here, and then these are both SH2 domains. So this
Yeah.
still binds to the phosphate. So yeah.
So like this is the SH2, and then when it like unbinds, this SH2 can then bind over here, then this SH2 can bind over here. So they like,
So they're the same site, but like you can kind of tell these invert relative to each other. So this one, or I guess this one in the diagram flips around so they can use those SH2s on either side combined to the same phosphate.
Alright, you got it.
Oh, there's
Oh.
more.
I think
Oh, but
Oh,
if you hold
yeah,
anyone-
you got it.
All right.
Cool. Or
Okay.
in case you guys need it, it's like written out.
Okay, so GPCR is our last pathway that we're gonna cover. I guess I would go with that. So we have a better diagram
Yeah.
on the next slide. This is the one that Laura uses, and this is, I guess, the one I need to explain it. So for this one, we have our GPCR, which is our G protein coupled receptor. It has 7 transmembrane domains, so that is these regions. They all kind of go back and forth through the membrane. The whole thing is the receptor. And the reason it's called G protein coupled receptor is because this is our heterotrimer of G proteins. So they're all proteins in association with the receptor, kind of like the JAKs are with the prolactin receptor. And we have G alpha, G beta, and G gamma. So when the G protein coupled receptor receives a ligand or a signal, I think they call it a ligand in this diagram, that will stimulate G alpha to drop its GDP, pick up the GTP because it'll just diffuse into the binding pocket. And then our G alpha will dissociate from G beta and gamma. G beta and gamma will stay in a complex. So we'll have alpha that can go do its own thing, stimulate a pathway, interact with the effectors, whatever, on its own. And beta and gamma will stay together and interact with their own effectors. And then we have RGS, which is another regulatory protein. And it will cause G alpha to speed up its intrinsic rate of GTP hydrolysis. And so it will hydrolyze the third phosphate in its GDP. It will become GDP, then is inactive, and it rejoins the GPCR along with beta. So an important note about RGS is it is not an enzyme. It's not cleaving that phosphate itself. It's just stimulating G alpha to do that on its own. Kind of like we talked about gaps and gaps. The GPCR and the RGS would be the gaps and gaps for this time for G alpha. Do you feel okay with that? Am I going too fast? I want to make sure that you're going. Okay, so this is just, I guess, a better depiction of that. The only thing about this diagram, this is supposed to be our GPCR, but in reality, they look a lot more like the one that Laura had on her diagram, where it's kind of like this one goes back and forth through the receptors to those 7. So that's pretty exciting. These 7 different memory regions, so that would be in addition to that diagram. Okay. Oh yeah, and this is what I just mentioned. So GEF is not an enzyme. It promotes GTP dissociation, and then GTP is present in higher concentrations in the cell, so it diffuses in. And GAP also not an enzyme. It binds to the GTPase, which in this case is our G alpha. And it stimulates its intrinsic rate of GTP hydrolysis. So it on its own, the alpha on its own, can cleave between the second and third phosphate to bring it back to a GTP bound inactive state.
I'm gonna be sitting, but I'll be using like my pointer and everything to point out anything that you guys might need to know. And again, like just like Lily's been doing, if you guys have any questions, feel free to just stop me whenever. So we're gonna go into the cytoskeleton, microtubules, and all of that. So the 3 types of cytoskeleton that the cell can have is actin, intermediate filaments, and then microtubules. We of course focus on microtubules the most, but it's good to know about actins and intermediate filaments as well. The actin is mostly used for muscle contraction, cell mobility, cytokinesis, things like that. And then intermediate filaments help maintain the shape of the cell. These are really important just obviously for cell function, but especially for like cell division, it's pretty important. But you guys don't need to know too much about like what they do molecularly because I don't think we really cover it that much. Microtubules, however, we definitely do need to know about. So they help resist compression. They do like the whole push-pull mechanism that we talk about to try to get chromosomes and spindle poles to the opposite ends. And that's really crucial for cell division as a whole. All right, so starting off with spindles, spindles are microtubules that microtubules form a spindle and separate chromosomes during the M phase. So these are your spindle poles. And then you have a bunch of different types of spindles that you can have. You can have aster microtubules, which are these green ones. I think of aster as like asterisk, and like an asterisk is like a star. So they kind of look like stars kind of going out in all directions of the spindle pole. So they grow from the centrosomes and they attach to the cell cortex. And then you can have kinetochore microtubules. These are the ones that are shown in blue. They grow from the centrosome as well, and then they go towards the kinetochores of the chromosomes. And then you have lastly your interpolar microtubules, which are also growing from the centrosomes, but then they overlap at kind of the metaphase plate area. They overlap spindles, and then they have, what's it called? Motor proteins and different things like that to kind of hold them together that we'll go into a little bit later. All right, so the structure of a microtubule, you have tubulin heterodimers. So you have your alpha and your beta tubulins. A microtubule, they're not charged per se, but they do have a positive and a minus end. Tubulin addition can occur on either end, but it's definitely more likely for them to occur at the plus end. So if you think about a cell, the negative end or the minus end is going to be embedded within the centrosome. Sorry, not the centrosome, the centromere. And the whole microtubule is made up of 13 protofilaments. So each one of these like long lines is a protofilament, and then 13 of them form a wrap around to make like a hollow tube in the middle. And then the microtubules extend from the microtubule organizing center. So that's just shortened to MTOC. So if we go back to like this picture, the MTOCs would be here at the spindle poles within the centrosomes, and then they grow outwards. So this would be the minus end, and then over here would be like the plus ends. All right. So just kind of going into kind of how they grow and shrink a little bit more. When you're in the cell, you don't have any growth, pretty much no growth or shrinking at the minus end, because the minus end is inside the nucleating site or your MTOC. In this case, it is the centrosome. And so obviously if it's embedded within the centrosome, it's not going to be able to kind of like unembed itself. So you're not really going to have any growing or shrinking. All of the action is mostly going to be at the plus end, because your beta tubulin, specifically your beta tubulin, have the ability to hydrolyze. And so that's what's causing like the GTP and GDP able to grow and shrink. However, when you're in vitro or in the lab, you don't really have a nucleating site unless you obviously build one, but you have the ability to kind of grow and shrink on both ends. It's just that, again, the plus end is a lot faster. And so the things that you need to grow microtubules in vitro is you need pure tubulin and then GTP in a test tube. It's good because you have a more controlled environment, but obviously you're doing it in a tube, so you don't know obviously what the cell environment would actually be like. So whatever you do in a tube, it's not positive that it'll translate into a cell. All right. So just to explain a little bit more about tubulin so that you guys understand kind of how the tubulin works and its mechanism. Again, it's a dimer and it's the monomer of microtubules. They also are a GTPase, so they act on GTP and GDP. Your tubulin is, again, a GTPase. They hydrolyze GTP to GDP. When it is GTP bound. They have a better affinity for each other. So that's when you're able to grow, they're able to bind onto each other and continue growing. But when the GTP gets replaced with the GDP, that's when they don't have an affinity for each other. And then that's when catastrophe can happen and you're just going to have a shrinkage and depolymerization. And yeah. All right. So microtubule dynamics. This is kind of specific to in vitro. So we have like this very particular or commonly occurring graph that we've been seeing all throughout class. So the way that it works is it's you put like again a bunch of tubulin and GTP into a tube, and then you use a spectrophotometer. I don't really know how to pronounce it, but spectrophotometer, thank you. But essentially it calculates or takes in how much light is absorbed. So yeah, you have absorbance on your y-axis and then time on your x-axis. If you choose, or if a question comes up on the exam where you have to like draw one of these graphs, I'm pretty sure it's important to label your x and y axes. Laura typically likes to look out for that just so it shows that you understand what the graph is showing. And so because it shows absorbance, the more microtubules you have present, the higher your absorbance reading, because the more stuff you have, the more light that's being absorbed. So that's why the longer the time goes on, the more microtubules that are growing, and then the higher your absorbance.
I can just add something.
Oh yeah, go for it.
So I don't think she said this with this class, but when I took her class, one of the things like for the drawing questions for the polymerization assay, one of the things that she was really particular about is when you start with all of your stuff in your test tube, you don't have any microtubule formation yet. So you're starting at the origin if you were to draw this because you have no absorbance. So like if you're starting halfway up, that would not be correct because you don't have any microtubule formation yet. I don't think she said that.
I guess that's part of, yeah, I don't think she said it either, but then that's why the lag phase is so important. So when you draw these, you got to draw the lag phase. Don't skip the lag phase. Speaking of, good transition, you have 3 phases that are typically seen in the in vitro assay. So you have your lag phase in the beginning, and then you have your elongation phase in the middle, and then plateau phase. So I'm assuming what you're saying, Lily, is like sometimes some people draw from like the middle.
Yeah, like people start like at the 0.2 mark instead of the 0.
Oh, like they'll draw like over
Yeah,
here. Okay, yeah. Don't draw from over here. You got to draw from 0 because there's like literally nothing in your tube is what you're starting from. And so if you start with nothing, then there's no absorbance. So then it's got to start at 0. Cool, thanks. All right. So for the lag phase, it takes some time for the formation of seeds and microtubules to start because like Lily said, you just threw a bunch of free tubulin in. They can't just grow from nothing. They have to take a little bit of time to kind of come together. So you start off with like pure tubulin, and then you start to form your dimers. So like your alpha and beta tubulin dimers, and then those come together to form oligomers, and those come together to form those long protofilaments that we saw earlier. And then you have a sheet of protofilament, which again is 13 of them. That sheet closes and you can then form a seed where it's kind of like a hollow tube now. And then from there, you can finally start to elongate, but all of this takes time. And so that's kind of why you have a lag phase. If, say, for instance, you added a bunch of seeds in already with the free tubulin, then maybe, and then Laura was like, oh, how would your graph look like then? You wouldn't have a lag phase, or you would have a much shorter lag phase because you didn't need to spend that time forming all the seeds, if that makes sense. And so, yeah, and also on that same note, the higher amount or the more concentration of free tubulin you have, the shorter your lag phase, because if you have a lot of free tubulin hanging around, it's just a lot quicker for them to all find each other and form the microtubule faster. So if you have more tubulin, you're going to have a shorter lag phase. If you have less tubulin, you're going to have a longer lag phase. Okay. Oh, I think I kind of answered this already. But if you had stable aggregates or seeds already provided, for example, gamma tubulin ring cells, how would your graph look like? Awesome. Yeah, so you would have no lag phase, or at the very least, a much shorter one. Oh, there's more. Oh, just kidding. This is just the answer. Nice. All right. So elongation phase is kind of like the middle part of the graph. So at this point, you have small microtubule seeds that are attaching to each other, and they're going to be able to grow and form a larger microtubule. As soon as because it's exponential, because as soon as you have that seed forming, it's going to be able to rapidly add tubulin, just because of the high affinity that all of the positive beta side, the beta tubulins have for each other, where they're all GTP bound, they have a high affinity for each other, and they're all going to be able to add and grow rapidly. Again, primarily to the plus end. And so the plus end, oh, sorry guys, I am saying information, but I didn't like look through these slides, so I'm kind of repeating myself. But the plus ends GTP have a greater affinity because, again, they are GTP bound. They have a good affinity for each other, both horizontally and longitudinally. And then that's all part of why it's growing really rapidly. But of course, as you grow longer, you are in a tube, and it's not like you have a never-ending supply or surplus of free tubulin. And so as your microtubules elongate, the amount of free tubulin you have decreases, and so your rate of assembly is going to slowly start to decrease as well, which leads us to the plateau phase, which is the last phase of the cell. So basically, the plateau phase doesn't mean that your polymerization has stopped. So that's a common misconception. Instead, what it means is your rate of microtubule assembly is equal to the rate of disassembly. So basically, you have like an even distribution of amount of microtubules being added and the amount that are depolymerizing on the other end as well. Eventually, though, your GTP hydrolysis is going to start catching up. So if you can picture it in your head, on the plus end of your like tube or test tube, on the plus end, you have a bunch of polymerization happening, but then on your minus end, you kind of have GTP hydrolysis going forward, and they're exchanging the GTPs and hydrolyzing it for GDP. And this is going to cause depolymerization because GDP does not have good affinity for each other. When they eventually are all GDP bound, it's going to cause a catastrophe. They're all going to fall apart from each other because there's no GTP anymore for them to stay together. Oh, okay. So I think this is like a series of questions. In an in vitro, say what happens after hydrolysis completely catches up or hydrolysis is stimulated.
That's it.
What happens?
Or is it a catastrophe?
Yeah, exactly. So after it completely catches up, it's all GDP bound. And so in a in vitro assay, that's a problem because you don't have any more GTP that are free unless you add more GTP. Otherwise, it's just going to catastrophize. I don't know if that's the right grammar. Okay. Oh. So question 2, how would you cause microtubule polymerization and microtubule depolymerization? I don't know what kind of answer it was. Oh, okay. So say for instance, if you were in like an in vitro cell or in vitro tube, and you've used up all your GTP, now all of them are all hydrolyzed too, but you wanted it to keep growing, what's something that you could do to cause more growth? Yeah.
Like add tubulin GTP.
Yeah, exactly. So you could add more free tubulin GTP. What about the opposite if you wanted your microtubules to start shrinking? Yeah, so we'll remove what? You could either remove and like decrease the amount of GTP, or you can also just, what we talked about, remove the GTP cap. I think something we've talked about too, and like one of the problem sets is cutting it in half, because remember, like as you're growing, you're also hydrolyzing. Most of the microtubule is most likely already hydrolyzed. So if you cut it in the middle, you're going to expose a GDP area, and so it's all going to fall apart. All right. So dynamic instability. The main idea with dynamic instability is that microtubules can grow and shrink independently of one another, and it's really important. So again, growing doesn't mean it's a good thing. Shrinking doesn't mean it's a good or bad thing. You want to have growing and shrinking for a nice and healthy cell that can divide. And then whether or not the microtubules are growing or shrinking is dependent on the rate of GTP hydrolysis. When you have GTP hydrolysis, you're going to be depolymerizing because you have GDP. When you have tubulin addition, that's going to be polymerizing because you have GTP. So if you have more hydrolysis than the amount of tubulin, you're going to be shrinking because it's going to be unstable. There's not going to be a GTP cap. But if your rate of hydrolysis is less than your rate of addition to additional tubulin, then you're going to be growing. So this is just an example of a growing one. Your rate of things being added is faster than it can catch up. So you're going to be growing. But in this scenario where it's shrinking, your rate of hydrolysis is faster than the amount of tubulin being added on. So that's why it's going to be shrinking. Again, if I'm going too fast, let me know. A very good video that does not seem to be visible. We'll fix that when we send out the slides.
It is on like our version. See if it's a
Oh,
transition.
it is a transition. Okay. That does not play. Okay. Well, either way.
This is the one that Laura showed in class.
Oh, okay. Well, either way,
Oh wait, no, mine is a different.
or you different slides, right?
Okay, but this one is still here. It's a picture.
Oh, okay. Well, it's a link. So once we like send out the slides, you guys can watch it for extra support if you need like a more visual of what's actually going on, type of like an animation. It's always good. All right. So we've talked about there's a bunch of different drugs that can affect microtubules, but the main three that we talk about for this class are Taxol, colchicine, and vinblastine. So it's definitely important to just kind of know what each one does, because sometimes some questions that she can put up are, oh, like if you add colchicine, what's the graph going to look like now? If you add Taxol, or if you like take away Taxol, what's it going to do to the cell? Things like that. So what Taxol does is it binds and stabilizes the microtubule. And when you stabilize a microtubule, that means you're going to have very little or no depolymerization. Basically, it prevents like hydrolyzation from happening. And so this can be used as a chemotherapy because, again, in order for cells to divide, you have to have growing and shrinking of microtubules. And if you're not able to shrink, in this case, with no depolymerization, then you're not going to be able to divide. And so obviously that's a good thing for cancer because you have too much division. But in colchicine, it's going to be able to bind and prevent polymerization. So they're kind of the opposite of each other. So this is a microtubule destabilizer, and it prevents tubulin from being added on. This is more used as like an inflammatory control usually within medicine. And then vinblastine, you can bind it and prevent polymerization as well. So these are similar. They both prevent polymerization and they both destabilize. I think she talks about these two more, but it's definitely good to know about all three. Okay. So you add colchicine to your in vitro assay at time equals 0. What results would you expect to see? So colchicine destabilizes the microtubules. So then what would you probably see? Yeah, so you would probably get no growth. Sometimes some students draw like a little bump. That's fine too, because it's kind of like to show the ambiguity in biology where like anything can happen. But as long as you explain yourself and explain like why you either drew like nothing or a little bump, that's fine. I drew these graphs on Word like my sophomore year, guys. It was really tough. Okay. maps and motors. I think this is the last part of this section. Awesome. So MAPs stand for microtubule associated protein. An example of MAP that is kind of the only example really that we talk about is tau. So tau is highly expressed in neurons within the central nervous system. So you can find a lot of them in like the brain and things like that. They bind to microtubules and they stabilize it, which if you think about is really important because, you know, in your brain where you want things to be stabilized, you don't want your microtubules to suddenly catastrophize, or I keep saying that, I don't know if it's a word. You don't want your microtubules to suddenly depolymerize and have a catastrophe where if you would imagine like your microtubules are the highways, and if you have things that are moving around on them that are trying to get to important places within your brain and suddenly they like give out under you and they fall, would not be a good look for your brain. Okay, so motors are where we talk about them a little bit more. There's a lot of different types of motors, but they hydrolyze ATP in order to move along the microtubule. There's a really good movie in like the movies folder in Moodle, I think. If you guys haven't looked at it, I do recommend checking it out, especially if you're like a little bit more of a visual learner. But they hydrolyze the ATP in order to help move the microtubule along, or move along the microtubule. Basically, like these little heads kind of act as like feet, and then the ATP, when it hydrolyzes, it can let go of it, and then it adds onto ATP, and then it like sticks on again, lets go, sticks on. They kind of like walk along the microtubule literally. And their main job is to transport cargo around the cell via microtubules. So an example that we often like to use is that the microtubule itself is the highway, and then the motors are your cars transporting cargo or whatever it needs to transport. They do have directionality, so that's important to know, especially when we get to like cell division. But your kinesin generally walk towards the positive end of the microtubules, except for kinesin 14. That's an exception. They walk towards the minus end. They are also more diverse. They have a lot more types of kinesins, and they have a bunch of different functions. Your dyneins, they walk towards the minus end and are a lot more common in like flagella and cilia, but they are present in our cells as well. Okay, so this is kind of like an overview of all the different types of kinesins that you should either know about or just like at least understand what they do and which direction they walk in. So kinesin 1 is a dimer that walks along the microtubule. Again, this is plus end walking, so it's going to walk towards like the middle of the cell if you're thinking about like mitosis and things like that. They primarily transport cargo around. Kinesin 3 is also a dimer that walks along the microtubule, also plus end directed, and they also transport cargo around, specifically in the neurons. Kinesin 5, also known as EG5, is a specific example. This one's a tetramer, so that means that instead of it just looking like this, they have like 2 heads and 2 tails all kind of together. And then they also walk in the plus end directed on the microtubules, and they help bind the interpolar microtubules that we talked about earlier slides ago, to kind of connect them and keep them together. And they're definitely required for proper spindle assembly. And then kinesin 13 is a dimer, but they don't walk processively on microtubules. Instead, they kind of just like diffuse along the microtubules towards the plus end, and use ATP hydrolysis to destabilize the plus ends. So an example of a kinesin 13 is MCAC. They're used during like anaphase to really try to shorten the microtubules and get the chromosomes towards the opposite ends of the cell. All right, this is just kind of a depiction of all of them. An example of EG5 again, like they're tetramers, so you can see that they have 2 heads and 2 tails rather than just like here tail, here head. All right. How does kinesin 5 or EG5 push spindle pulls apart, if you guys remember? All right, so they are plus end directed. Again, remember kinesins are plus end directed. And so when you walk towards the plus end, so if this is the plus end, you're walking towards it. These are the egg fives. As you're walking towards the middle, you're kind of like dragging the microtubules behind you. And as you're dragging it behind you, then your spindle poles get pushed further apart. So think of it like as you're walking, your feet are like kicking behind you. And so you're pushing the spindle poles apart from you, away from you. So that's kind of like one way to remember it. I know it's like tricky to think of, but. Okay. This is also another video. Feel free to take a look at it if you want later on. But again, there's a lot of good videos of like kinesins walking or motors walking in the movies folder. Okay. Any questions about microtubules, motors, maps? Sweet.
So I'm gonna cover the first part of the cell cycle. I think this one's a little bit of a shorter section. So just a quick overview of the cell cycle. I'll go through this pretty fast because we'll talk about this more in depth. But G1 is our first official stage of the cell cycle. It's our first gap/growth phase, however you want to think about it. So what the cell is doing here is it's preparing for replication, the next phase. And that means it's making sure it has enough nutrients, like cell machinery, protein, whatever. And obviously it's checking for mutations the entire time. And then it's not marked on this diagram, but towards the end of G1, we have the restriction point, or it's called abbreviated R, it's called start and leaves, and that's the point where the cell commits to the cell cycle. So after it passes the restriction point, it's either going all the way through, it's dividing, or it's dying because it gets stuck in one of the phases for too long. And S phase is our synthesis phase where we replicate our DNA. Again, we check, we make sure this, in a lot of textbooks, isn't an official checkpoint, but it's still making sure the DNA has any mutations. G2, we are preparing for chromosome segregation, and we have another checkpoint at the end. Then mitosis, we have our prophase, metaphase, anaphase, telophase. It ends in cytokinesis, and then two daughter cells separate from each other. And we will talk about the anaphase checkpoint. The only other stage that we kind of have removed from the cell cycle is G0. And the cell hangs out in G0 if it's waiting like for another growth factor signal to restart the cell cycle, or if it's just removed from the cell cycle, if it's permanently done dividing, whatever. This means that it's not going through this loop of replication and division. Okay, so CDKs are super important. They are our master cell cycle regulators. That's what Laura calls them. So they are the thing that is pushing our cell through the cell cycle. And they are regulated by cyclins, which we'll talk about in the next slide. And phosphorylation, dephosphorylation, WEE1 is an example of where we have deactivating phosphorylation. So if you really think about phosphorylation as a on switch, it is not always. And CDK inhibitors. So to activate our CDK, we need an activating phosphate and sorry, our cyclin that corresponds to whatever phase the cell cycle we're in. And when we have our checkpoints where we're checking for mutations, for damage, for any of those issues, for like not having enough nutrients, or not having enough proteins available, the way that we kind of put the brakes on, slow down, halt the cell cycle is by downregulating the CDK. So the thing that's really good about having one cell cycle regulator is obviously that we have all of these proteins that are involved in all of these different phases of the cell cycle. But because we have CDK as our central regulator, instead of having to target all of those proteins individually when we want to downregulate, we can just target CDK, and then that will stop everything below it. So depending on the phase of the cell cycle, depending on what it's doing, CDK can activate different substrates or deactivate. And it gets its binding specificity from the cyclin that it's bound to. So that determines what it is. So cyclin, like I just said, is the thing when it's bound to CDK, it's going to return what it binds to. And for our different phases of the cell cycle, we have different cyclins. You don't need to know what they're called. It might be helpful to draw this diagram. I had it drawn. But cyclin levels vary between different stages of the cell cycle based on like, what needs to be bound to CDK and what CDK needs to bind to. That's determined by what cyclin it's attached to. And cyclin is only regulated by at the transcriptional level and then by degradation. So when we want less of a certain cyclin, we don't like inhibit it, we just degrade it. And that is in the proteasome through ubiquitin. Okay, so when we have our cyclin CDK complex, our CDK has an activation loop, and that is the region of it where like we have kinase activity, where it's phosphorylating. But that is tucked, so you can kind of see here, inside of the CDK protein. So first, we need to bind to our cyclin, and that is going to expose our activation and kind of bring it out from within that protein. So we have our activation loop, we have our kinase region exposed, but it's still not active until we have a CDK activating kinase to phosphorylate right here on the activation loop. And then we're officially active. We can go and regulate our downstream effectors. The opposite of that would be the CDK inhibitors. So that is one of our mechanisms of downregulating CDK activity. And there's other proteins that can prevent CDK from phosphorylation. Okay, so when we have cell stress, our really, really important transcription factor that we want to remember is p53. So anytime that we have mutation, any kind of stress that we need to pause the cell cycle for, p53 is a tumor suppressor. Do you guys know what that means? A tumor suppressor is the opposite of like a proto-oncogene or an oncogene. I don't remember. Can you talk about both? I feel like we only use either the word proto-oncogene or oncogene.
Yeah, the only difference is like a proto-oncogene has the ability to become an oncogene,
Right.
but you don't need to know.
Yeah.
So that's fine.
I don't remember which word we use for that here, but a tumor suppressor is the opposite of an oncogene or proto-oncogene, where it's going to stop the cell cycle or prevent the cell from dividing. So p53, our transcription factor, is going to upregulate p21 transcription, and p21 is the CDK inhibitor, like we talked about in the last slide. So when we have CDK inhibited, we can't push the cell through the cell cycle, which means that we're not going to have division. So if we have damage, if we have a mutation in our DNA, something like that, p53 upregulating p21 transcription will halt to prevent us from going through the cell cycle. And if we need, if the damage is like so bad that we can't fix it, then it can stimulate apoptosis and kind of like cyclins when it is not needed or when it's inactive. Well, I answered this. But what happens if p53 is no longer available? Or what would happen if we had some sort of cell stress DNA damage and we didn't have p53? Okay, so if we had DNA damage and we didn't have p53, then it wouldn't be able to upregulate p21 translation, which means that we would continue through the cell cycle even with that damage, and that could possibly lead to like a cancerous phenotype or just the cell dividing when it's not supposed to. With damage, it might cause, you know, a permanent mutation, something like that. Okay, so G1. Like I mentioned before, it's checking for nutrients, it's checking for size, it's checking for availability of like cell machinery, it's checking for DNA damage. And those are kind of going to be consistent themes throughout the entirety of the cell cycle, especially checking for DNA damage. We're going to be doing that constantly. We're really going to want to divide conclusions. So Laura does distinguish between early, middle, and late G1 as kind of some phases. So I think it's important to know that, especially in relation to pre-RCs and pre-ICs. So during early G1, we have inactive G1 CDKs and G1 CDKs, G1 cyclin CDK, abbreviated version of that. So before our G1 CDKs are active, or our G1 cyclin CDKs are active, we form our pre-replication complex, our pre-RC. And then during mid to late G1, we have our restriction point. And like I mentioned before, that is the point where the cell is committing to the cell cycle. It's saying like, all right, we're ready to go. We're big enough. We have enough nutrients. We don't have damage. And after that, it either has to divide or undergo permanent cell death. And late G1, we have full active CDK or G1 CDK activities, and we can form our pre-ICs. So a little more on pre-RCs. There are a couple important proteins. We start off again during early G1, so we don't have a lot of CDK activity, and that is important. And the first thing that we do is we have the origin recognition complex, ORC, which finds the origin that we're dividing from. It finds, and then it can recruit CDC6. And this ORC CDC6 complex can recruit CDC1 and MCM, helicase. And in this diagram, we see we have multiple MCMs because they are helicases, so they can unwind DNA in both directions, like we see from our replication model or replication forks, how it goes bidirectionally and zooms both ways. And then in our mid to late G1, we pass our restriction point if we're ready to. So what happens when we commit to the cell cycle is we have these proteins called RB, which stands for retinoblastoma, and E2F. E2F is a transcription factor for S-phase proteins. But right now it's being sequestered or inhibited by RB. So when we have our active cyclin CDK complex, then that can go through the kinase activity of CDK and phosphorylate RB on two positions, and that causes a conformational change that makes it lose its affinity for E2F. It lifts off, and then when E2F isn't being sequestered anymore, it can go and act as a transcription factor and make our proteins for S-phase. So once we have that in motion, that's when we're committed, we're dividing no matter what. Okay, so now during late G1, we form our pre-IC. So a pre-IC is just like a pre-RC, but with more stuff added onto it. But we, let's see what I have. We make our pre-ICs when we have high CDK activity. So the reason that is important is because during our early G1, when we have really low CDK activity, that's when we form our pre-RCs. That's when we have to form our pre-RCs, because if we start that process late and we have high CDK activity, then all of these pre-IC proteins are going to be recruited early, and we won't be able to have pre-RC formation occur correctly once we have that high activity. But now that we're in late G1, now that we've committed, now that we have high cyclin CDK activity, we can recruit all of these extra proteins onto our pre-RC. And then DNA polymerase will be loaded, we'll start replication. And as soon as we start replicating our DNA, that is when we officially enter S-phase. And that's when our origins fire, which just means when replication starts from the origin. Like we saw in that one diagram from class, you can have, or actually you do have multiple origins on the DNA that all kind of open up. So this is happening in multiple places on all of your strands. Okay, so a couple of true or false questions. True, give me a thumbs up or thumbs down. In response to DNA damage, cells should increase concentration of CDK inhibitors. True or false? Okay, yeah. So I'm seeing mostly trues, and that is true because CDK is our protein that pushes the cell through the cell cycle. And when we have damage, we want to halt our cell in the cell cycle. We don't want it to divide further. Especially we don't want to make daughter cells that have or carry that damage. So the best way to stop the cell cycle from continuing is by inhibiting the master regulator that's going to push it through the cell cycle. All right, so true or false? In response to DNA damage, E2F is active. Some multiple falses. From the people who responded, that is correct. So like I just mentioned, our RB E2F complex. In that complex, our E2F, when it's active, when it's unbound from RB, is a transcription factor for S-phase proteins. And once we have those proteins, we're essentially like getting ready to start dividing. We're moving the cell towards S-phase, so we do not want E2F activity when we have DNA damage. We want RB to be active and bound to E2F to keep it sequestered to keep it from pushing. So on a similar note, could E2F be a cause of cancer? True. Yes or no? Yeah, I'm saying yes. Does somebody want to say why?
If you can't inactivate, we'll just keep producing, keep the product cells.
Right, yeah. So if we have overactive E2F, then we're going to be producing these S-phase proteins that we don't want. We don't want to move to S-phase. We don't want to push forward. We don't want to pass the restriction point. So sometimes we need to downregulate it. And can a cell divide if E2F does not function? Yes or no? Yeah. So does somebody want to say why? Go ahead.
It'll never like commit, it'll never start like the proteins for
Right, yeah. So E2F is a part of our restriction point, like set of proteins, and we need to pass the restriction point for the cell to commit to division. But if we don't have this protein that makes our S-phase or transcription factor that makes our S-phase proteins, then not only can we not pass the restriction point, but we also don't have the proteins that we need for S-phase. Last one, I kind of talked about this, but true or false, pre-RCs can form at the end of G1 after CDK is fully activated. Yeah, I'm seeing false. Does somebody want to explain why it's false? This is kind of tricky, but I did mention it. Either one of you go ahead.
I have what I remember it was like only in late Q1, and it's when there's like really low levels of CDK.
Okay, so I'll go over that a little more. So during early, you are on the right track. During early G1, we have low levels of active cycling CDK or G1 CDK. So when we have low levels, that is when ORC will recognize our origin. We'll have CD6, MCN, CD1, all of those form our pre-RC. Then later, during late G1, we have really high G1 CDK activity. And when we have high activity, that stimulates or promotes, I guess, the pre-IC proteins to go and bind at the origin. So if we have pre-RCs trying to form at the end of G1, ORC would still be able to recognize the origin. We would still be able to recruit CDC6, MCM, CDT1. That would all still work. But because we're at the end of G1 and because we have high G1 CDK activity, at the same time, we're also going to be recruiting pre-IC proteins when the pre-RC is not ready for those to add on yet. So we would have a whole bunch of proteins coming in like before it's time, before it's ready, because we have that active CDK. So we need to form pre-RCs before we have fully active CDK. Does that clear up? Okay. Does anybody else have questions on that?
Any questions on cell cycle 1, anything Lily talked about in general? All right, well, cell cycle 2 is also relatively short, I think shorter than cell cycle 1 as well. So it shouldn't be too bad. We're almost done, guys. All right. So CDK inhibits new pre-RC formation. So we kind of ended off with the end of G1. Now we're going into S phase, where DNA is going to get replicated, but obviously you don't want pre-RCs or more pre-RCs to form, because now that you've already fired, you don't want to have like double replication. And so the way that that's going to happen is your CDK is going to phosphorylate a bunch of things so that your pre-RC is not going to form again. And so obviously, in order to make sure your pre-RC is not going to form again, you want to get rid of all the things that make up the pre-RC. So one of it is phosphorylation of the ORC, because if you phosphorylate the ORC, that inhibits CDC6 from binding. So if you get rid of the ORC, CDC6 cannot bind to it. That's already one step out of the way for your pre-RC to form. You can also phosphorylate the CDC6 itself, and then that's going to lead to ubiquitin-mediated proteolysis. That basically just means it tags it with ubiquitin, sends it off to like a proteasome or something, and then just like not kills it, degrades it, yeah. Which I guess that's what it's kill. And so the last one is CDK phosphorylation of the MCM complex. You're going to phosphorylate the MCM complex, and it's going to be exported out of the nucleus with exportin. And this is important because these three make up the pre-RC. And so if you get rid of each one, you're not going to be able to form the pre-RC. What's important to note, though, is that obviously MCM that is currently like in the process of being used for DNA replication, those are not going to be phosphorylated because the cell needs it. But any like extra MCM that might be around in the cell, those are going to be kicked out of the nucleus. All right. So now we are in the S phase, and you're going to have cohesin rings that are going to be forming around DNA that has been replicated at this point. So you have your sister chromatids, and you're going to have your cohesin rings around all of the sister chromatids. This is so that you can keep the DNA strands together. They're not gonna, it's basically like another measure for the cell to make sure that they stay together when they need to be together. It prevents chromosome segregation during spindle assembly. So you don't want to segregate your chromosomes before the cell is ready. That would cause a lot of trouble. All right, so after the S phase comes G2 phase. And so now your cell's kind of getting ready for chromosome segregation. And there's three main things that it's going to be checking for. It's going to be checking for DNA damage, whether or not your replication was complete, because it would be really detrimental if your S phase replication hasn't been done yet and then you already move on. And then it's also a time for a little bit more growth, for the cell to grow a little bigger, get more nutrients, whatever it needs to be able to go through the M phase. So specifically in the G2, you have a G2 checkpoint. Here, your M cyclin levels are going to be rising. But even though your M cyclin levels rise and they can bind with mitotic CDK, when they bind together, that's still an inactive MCDK. The only way that your complex is going to become active is if you have phosphorylation. So first, you're going to have CAC, which is an activating kinase. It's going to phosphorylate in this picture down here, but it doesn't necessarily have to be like at the bottom. So your CAC is going to phosphorylate M-cyclin CDK with an activating phosphate, but at the same time, it's going to be phosphorylated by an inhibitory one as well, by WEE1. And so now you have an inhibitory phosphate and an activating phosphate. And so they kind of like in your mind, you can think about them canceling out, because if you have both of these on there, your CDK is still going to be inactive. So at this point, it's not going to be able to do anything yet. Is that a question? Or did you have a question? Oh, sorry. Okay, just making sure. So how is it going to become active then? Your CDC25, which is a phosphatase, again, I think Lily said before, it's kind of like the opposite of a kinase. It's going to be able to take off a phosphate. So right now, at first, your CDC25 is only partially activated by a different kinase, kind of like upstream, known as PIK1. And so this phosphatase is able to take off the inhibitory phosphate that WEE1 had put on. And once this inhibitory phosphate is off, now you finally have an active M-cyclin CDK. This causes like a positive feedback loop though. So your active MCDK goes up and further activates your CDC25. And then that means you have an even more active CDC25, and you can take off even more inhibitory phosphates. And then you're gonna have even more active MCDKs. And then that's just kind of like advancing the cycle into M phase. So what if there was a gain of function mutation in CDC25? What would happen? There's a couple different answers, I guess. Did you have a guess?
I was gonna say that it would like always be taking off the phosphate, and then but sometimes CDD needs that inhibited phosphate in order to like grow.
Yeah.
I think.
Yeah, so exactly. It might leave the G2 earlier than it should. So half the reason, or one of the main reasons why it has that inhibitory phosphate is so like it's kind of a pause for the cell to make sure and check that everything's good. But if you don't have that inhibitory phosphate there to pause everything, you might have a smaller cell than necessary. It might be less developed. You might not have all the nutrients that it needs yet. So it's not always a good thing. Nice. All right, so this is just like an M phase overview, kind of us just summarizing each part for you to look at if you would like. But we'll go into each one in a little more specifically. So starting with prophase, the MCDK controls M phase. Scratch that, I don't know what that means. So your active MCDK now, because your CDC25 had taken off the inhibitory phosphate, it's going to phosphorylate the lamins, phosphorylate the condensins, and also phosphorylate your MAPs and motors. So those are kind of like the three main things that it does in prophase. Your lamin is kind of like the outer shell of your nucleus. And so when it breaks it down, or when it phosphorylates the lamins, your nuclear lamina is going to break down and just like disperse throughout the cell. And so that allows all like your chromosomes to come out and do what it needs to do. Speaking of, your condensin is going to be phosphorylated, and that induces chromosome condensation. So that causes your chromosomes to kind of go from like spaghetti looking DNA to more like compact sausages. And lastly, you're phosphorylating your MAPs and motors, and that's going to facilitate mitotic spindle assembly, which we talked about a little bit before. So those are like the main three things of prophase. Some more main ideas. Lily talked briefly about like sequential phosphorylation. So basically phosphorylation needs to happen in a certain order. And a lot of it is controlled by the CDK. So say, for example, you needed to pause something that's further downstream, you can go to an upstream CDK and stop it from phosphorylating or getting phosphorylated. And then that'll stop everything else downstream because it's such a big regulator. Spindle assembly is when your existing microtubules, that's important. Your existing microtubules are all around the cell already, but they're gonna have to depolymerize first with an example, katanin. And then that's where like that whole question about if you just cut it in half, what's gonna happen? So katanin literally just kind of cuts all the microtubules up. They all depolymerize, and then they're able to quickly reassemble again into spindles as they prepare for chromosomes to start separating. And then, you also have your kinetochores. These are large protein complexes. You can't see them, but they're really complicated. They're more than just like a red dot that's shown here. So they're large protein complexes at the centromeres of sister chromatids. These are where your kinetochore microtubules spindles attach to. And lastly, your opposing forces. So this is like that push-pull model that we've talked about. Your spindle formation is going to require opposing forces in order to stabilize. That's kind of how the cell knows you're ready to divide as well, because it'll feel like that balance of pushing and pulling. So your kinesins, again, most of them are plus end directed, and then your dyneins are negative end directed. And then an example would be egg 5 at the interpolar microtubules, kind of like sticking them together. So this is egg 5. You have like this one might be coming from this side, and then this bottom one might be coming from the left side, and then your egg 5s are connecting them together. All right. So spindle assembly checkpoint, we've been talking a lot about like push and pull, and like it'll just, the cell will kind of feel when it's ready. So what does that actually mean? So this is your metaphase, anaphase checkpoint. Basically, once all your cells line up, how does the cell know when it's ready to separate the chromosomes? So if your cells haven't lined up the chromosomes correctly, this checkpoint is going to be activated and your cell cycle stops until it's ready to go continue. The steps that actually occur is you have CDC20 is released. It's going to go bind and activate APC. So this like blue little thing is APC. CDC20 binds to it, and now your APC is activated. What APC does is it's going to target your securin and separase complex, and it's going to degrade securin by ubiquitinating it again, so sending it off to like a proteasome or something of that sort. Now that your securin is gone and out of the picture, your separase is able to do a conformational shift, change its shape, and now it's also activated. And it's going to be able to go and cleave all of the cohesin rings that we talked about that had occurred in S phase. Cleave off all of the cohesin rings, and now your sister chromatids are free to separate from each other because there's nothing holding them together. Oh, also important to know, it's like APC is when it targets M cyclin for degradation. So we're kind of nearing like the end of the M cycle. So that's why your M cyclin CDK levels are beginning to drop. And we'll talk about why that's important too for like the completion of the cell cycle loop. All right. In anaphase, you have anaphase 1, not anaphase 1, anaphase A or anaphase B. Essentially, they all lead to your chromosomes getting segregated, but the difference is in anaphase 1, your chromosomes are pulled poleward. So it's kind of like going from the middle outward, whereas in anaphase B, your poles are being pushed and pulled apart. So in A, you're going from the center outward. In B, you're taking what's already at the edges and you're pulling them even further apart. And that's how, these are like the two mechanisms for how your cell is going to get divided. So yeah, in anaphase A, your chromosomes are pulled poleward. These can be done by minus end directed motors because they're walking towards the minus end. So they're pulling the chromosomes toward the spindle poles. They can also be done by your kinetochore microtubules being shortened. It's literally like something at the kinetochores that are like eating away at the microtubules and shortening them. And then your spindle poles are still in place. But in anaphase B, your spindles can lengthen. So your interpolar spindles, they can lengthen with your egg 5s, and then that's going to push them outward. Or you can have pulling forces directly on the poles, pulling them apart from each other. Right. And then the last part of M phase is your telophase and cytokinesis. So finally, your sister chromatids have reached opposite poles. And now you're going to start to form this cleavage furrow where like on both ends of it, they're going to start to cave inward and kind of like cleave itself to form two separate cells. This is known as a contractile ring, and it's going to deepen and deepen until eventually it hits all the spindles that are in the middle, and then finally split it like halfway down the middle. And so remember before when APC was talked about and it cleaved off the cohesin rings? Well, APC also, like we said, started to degrade the M cyclins. So your M cyclin CDK levels are dropping. When your M cyclin CDK levels drop, that means your spindles start to disassemble, your chromosomes start to decondense, and your nuclear envelope reforms around your chromosomes. So everything that happened from being phosphorylated in prophase, the opposite is happening so that your cell is going back to normal. And so that's why this is like a reset and like the end of the cycle, because once again, you have low CDK, and it's possible for your pre-RCs to form again. So hopefully you guys can see how like it's literally a cycle where everything connects to each other. All right, so just some questions, or does anyone have questions before we get into like these practice ones?
There was a question on the last exam that said if you had a mutation in V1 phosphorylation site, so CDK can't be phosphorylated in V1, what would happen? Like where it gets stuck?
What was that? One more time? Oh, okay. Yeah. Sorry, my leg is bleeding.
That one at the top there.
Mutation in the V1 phosphorylation in CDK kinase such that CDK can no longer be phosphorylated. What's the question? Would the following mutations cause it to get stuck in the cell cycle? CDK can no longer be phosphorylated.
You just pulled the answer. What number is that one?
It is 5E. Yeah.
Is Laura okay with us reading these just so like you can
think
hear
so,
it
'cause
from the
we've
horse's
already-
mouth? Okay.
Yeah.
So what Laura said for this one is if CDK cannot be phosphorylated by V1 kinase, then CDK can't be inhibited in G2 or G1 if the cell uses that mechanism. This wouldn't cause the cell to get stuck. It would cause them to go through G2 too fast and would result in small cells and other issues.
Yeah.
Yeah, no problem.
Did you want to talk about like why you thought yes? And
I
then,
thought yes, because I thought if you had CDK that was active, you couldn't even start the G1 phase really. Because I thought it would just get stuck at G1.
oh, okay.
Yeah.
So I guess like the tricky thing is thinking like when to start, what part of
Yeah.
the cycle to start thinking of. So in this case, if it talks about V1, or in any case of any of her questions, when she says like if something gets mutated, just think about like what it would normally do if it wasn't mutated, and then what the opposite effect would be now that. So normally V1 stops it from moving forward. Well, then the opposite would be if you don't have V1, then it's just going to keep going. So
Right.
when it gets stuck, does that make sense?
Yes.
Yeah. So that line of thinking instead. Thanks. Oh, that was quick. Yeah, me neither. Okay. So what if you had a loss of function in APC? What would happen to your M cyclins? Oh, that kind of answered its own self. So a loss of function in APC would cause your M cyclins to be degraded. But if your M cyclins cannot be degraded, what would happen? Would your cell cycle be able to finish? No. Do you want to explain why? No worries. So if you are correct, it would not be able to finish a cell cycle because what M cyclin does is it causes your lamins to degenerate and your chromosomes to condense, and what's the third thing? Oh, and it causes your spindles to break down. So if your M cyclins cannot be degraded, then it's just going to stay that way. Whereas you want them to break down because you want your spindles to reform. You want your spindles to basically you want your cell to go back to normal. Like you want your envelope to form again. So if your M cyclins are not degraded, you're never going to be able to fully complete the cell cycle. All right. What if the cell only had a minus N directed dynein? So just think about kind of like the spindle poles. Would you expect the poles to be going even further out, or would they kind of like come in and collapse on themselves, or? Yeah.
Would you be able to do like
No. That's okay. So anaphase A is when your chromosomes are getting pulled outward.
Isn't it like anti, like they're minus undirected, but like with the, it's like their plus end? Even though they're
Okay,
both
so
in
that
the
is
part.
an option. Yeah, I take that back. So that is an option. What about if you think about the spindle poles? So if you only had minus n directed, what would happen to your spindle poles? So minus n directed, if you want to think about they're moving towards your poles. So then remember when you walk towards one direction, you're kind of like dragging it in the opposite direction. So if your poles are on like opposite ends and you're walking towards it, you're like dragging it towards the middle. So your poles are going to collapse on each other. Yeah. So in theory, it could be that anaphase A, only anaphase A would happen. But if in the entire cell, all you have is minus n directed, then anaphase A might not happen either because you don't have you don't have other forces keeping the poles at the ends. Does that make sense? You need to have like both basically in order for anaphase to happen. So for 3, what if cohesin cannot be cleaved?
Then this is just how much is needed to separate.
Nice. And then what phase would you be stuck in? Nice. And then what if securin has a loss of function mutation?
Did the chromosomes deplete too early? Or the separate too early?
That could be an option, yeah, because basically securin and separase stay together. And if securin can't do its job, which is keep separase from doing its job, then separase is always active. So you can do anaphase, but basically separase is never gonna become inactive if there's no securin. So you'll kind of always be like stuck in anaphase possibly, or you might not be able to complete the cell cycle. Okay, what about if dynein is on the aster microtubules, would that be anaphase A or anaphase B? An example of A or B. Do you want to explain why? Well, you said A or B.
I said B.
Oh, okay.
And it would be, like pulling the spindles apart plus undirected, so like the pole is, that's like the minus and it's going towards the plus towards the cortex.
Yeah, like it's pulling them further towards the cortex. Nice. What if the dyneins were on kinetochore microtubules? Would that be A or B? So this is similar to question 2 or related to. Thumbs up for A, thumbs down for B. Yes, okay, so I see some A's. So if your dyneins are on the kinetochores, it's gonna be an example of anaphase A because kinetochore microtubules are attached to your sister chromatid. And so if it's attached to your sister chromatid and they're walking towards the minus n, which is towards the spindle poles, that's gonna be pulling the sister chromatids apart from each other. And so that's an example of anaphase A. Please don't freeze.
I think it's our last one.
Oh, awesome. All right. I think at the end of these slides, it's just a bunch of videos to help you out as needed. Lots of videos. But that's it. Do you guys have any questions? All right. Well, that's it for tonight. Thank you guys for coming. And if you have questions, obviously feel free to email any TA.
Also, because we did mostly content for this,